While many teens went to sports camps, these high school students chose a different kind of camp to study stem cells.
It's the next big thing.
In some ways, stem cell science is similar to sports.
It's controversial, it's exciting, it's got all the ingredients for something interesting.
And it's dependent on key players for success. In that sense, you could say stem cells are like the ultimate all-around athlete.
Stem cells are like no other cells in the body.
Imagine an athlete who could succeed at any sport. Scientists have the equivalent of that versatile star athlete in the stem cell.
Stem cells have the ability to become any and every cell type in human body. No other cell type can do that.
As a researcher and senior scientist at the WiCell Research Institute, Rupa Shevdi teaches students of all ages about stem cells.
The discovery of stem cells has completely revolutionized the world of science.
It's totally changing the way we think about medicine. And I was cruising along in my career as a neurobiologist studying the brain, and I went to see a talk on stem cells, and I came out of the talk thinking, wow, that was it. I was hooked. And here I am, 20 years later, very deeply involved.
Stem cells are unique in three ways. First, they are truly blank slate or unspecialized cells. Second, they can multiply for very, very long periods of time. Third, they can become any cell type in the human body under the right conditions.
Human embryonic stem cells were first isolated in 1998 by biologist Jamie Thompson at the University of Wisconsin-Madison. Researchers derive stem cells from excess embryos donated for research. They're called blastocysts.
Scientists will disrupt the outer layer, which is the trophoblast, to get to the inner cell mass. The inner cell mass is then cultured in a lab setting. The passaging is what gives them the immortality or the ability to multiply for long periods of time. One blastocyst has the ability to give rise to one human embryonic stem cell line.
It's revolutionizing the way that a lot of us will do research in the coming years. I'm a heart researcher, and the best heart cells for me to study for human disease are human heart cells. With stem cells, I can get these cells, grow them up into human heart cells, and then have as many as I want to study in the laboratory.
Human embryonic stem cells, because they have the ability and the potential to multiply for long periods of time, will always give scientists a large number of cells to do many experiments.
And scientists in any field can benefit from this new supply of cells.
Human embryonic stem cells can become any cell type in the body if scientists can give them the right environment and guide them to become that particular cell. It could be a nerve cell, a blood cell, a pancreatic cell, a heart muscle cell, or a bone cell.
The problem is, at this point, scientists can't consistently control the direction the stem cells will take.
The embryonic stem cells are trying to make everything in a higgledy-piggledy sort of mess of cells.
We start the cells down the road, and they sort of take off and go their own way.
Like a versatile runner who decides to race long distance when the coach really needs a sprinter. Stem cells have a mind of their own.
The trick is how to get them to be bone rather than heart, rather than neurons.
We don't know each of the thousands of steps to get there yet.
The hope is if we can find the right triggers, we can get pure populations of the cell types we want.
Even though scientists can't completely control them yet, stem cells have already given researchers a huge advantage.
Before the derivation of human embryonic stem cells, scientists used the tools and resources that were available to them. We were using mouse cells because there was nothing else available.
As a cardiologist, Dr. Tim Camp used mice to test treatments for human heart disease.
The catch is that if you look at a mouse heart and a human heart, obviously they're different in size, but they're also very different in their electrical properties. So the mouse heart goes about 10 times faster than the human heart.
Kind of like practicing basketball with a tennis ball and only using an actual basketball during the real game.
So if you want to know how a drug is going to affect a patient and you only study that drug in a mouse heart, you may get some very different results when you study it in a patient.
You have to have the right tools to get the right results.
Stem cells may be the right tools to create entirely new treatments that trigger your body to heal itself.
I think what you need to understand is instead of a drug, you're going to take a cell. Because your bodies are made up of individual cells, and in disease, it's often the cells that get damaged. And if you can replace the tissues that are damaged, you can have a hope for some kind of recovery. Or perhaps we'll give you a drug that can make your own cells regenerate, rather like the frog that loses a leg and regrows the leg out. How about that? If we could regenerate not a whole leg, but just part of your heart, say, if it was damaged. These are exciting ideas in regenerative medicine.
When you're adults, you may have access to treatments that do not exist today.
Scientists say there's a good chance we'll see applications of stem cell science in the next 10 years. But keep in mind,
I'd say that scientists are the worst people to predict things. I've been a scientist for 30 years. Some things I thought would be very easy have taken 10 to 20 to 30 years. Other things I thought would be incredibly difficult have taken 10 minutes in the lab. I think you can get very excited because potential is what it's about.
but stem cell science has had its share of critics.
You may have heard that there is controversy associated with human embryonic stem cells, and I want you to know that scientists do not take this controversy lightly.
As a scientist, the ethics are a problem. I've always worried about using embryos or using fetal tissue.
The issues are challenging issues to all of us.
IVF created a problem of excess embryos. These are embryos that were not chosen by the doctors to be implanted.
So it's been those, what we call surplus embryos that would be frozen indefinitely or discarded, that these embryonic stem cells have been derived from.
I personally have an issue if human embryos would be created for the purpose of research. I do not have a problem using blastocysts or embryos donated willingly by the parents for research.
And the way I do it is I balance suffering.
You see what these diseases can do to folks, and so that's always sort of in the back of your mind and keeps the research going, because you want to find better ways, because our treatments aren't so good now.
I think most people in 10 years' time will probably be using an embryonic stem cell that's not really from an embryo, it's derived from an adult cell that's been reprogrammed. And that's everybody's big hope.
But these adult cells, called induced pluripotent stem cells, are in the very early stages of research. So for now, it's necessary for scientists to rely on embryonic stem cells.
In time, these adult pluripotent stem cells will probably remove all the ethical issues.
We like to answer questions. That's our job. We try and understand things. There's much to learn. And any time we can get sort of a jump on the game, find a new way to explore it, we're excited.
The discovery of stem cells has completely changed my life, and it will probably change yours.
Science is so exciting. You can have a breakthrough like that suddenly changes the whole field. That's why I love doing science.
The science that we've been talking about is happening right now in labs all over the world.
You might say the big game is being played out in those laboratories. We don't know how it'll turn out, but you can bet it'll be fun to watch. This educational video was produced as a collaborative effort between WiCell Research Institute and the Stem Cell and Regenerative Medicine Center of University of Wisconsin, Madison.